welcome everybody. So you're at the right place. So this is a meeting for runners here. And the organizers really try to do every effort to promote the meeting, even sort of running marathons. So nevertheless, we also will have some magnetic resonance today with two fantastic speakers. And upon the consent of the speakers, we will record and upload to the ICERMBS YouTube channel. So today, as already mentioned, we have two fantastic speakers, Chuck Saunders from Vanderbilt University and Vladimir Lishansky from Gulf University. And with that, I hand over to Professor Tsai, who will introduce the first speaker, Chuck Saunders.
Hello. Hi, Chuck. Can you hear me? So yesterday I received a surprising email from Christian with an attachment. That is the first paper published by Chuck. And he said, well, he published the first paper with you. Would you mind introducing him? And so, of course, it's an honor for me to, of course, introduce my former student or colleague. And so that was 1988, and it was a JAX communication. I don't know if you remember what was it. It was actually quite advanced at that time. It was using deuterium NMR to study the dynamics of a substrate nucleotide bound to an enzyme. That's adenine kinase. So, so I reread that paper today, and it is still very, very nice work, Chuck. And of course, Chuck, you know, he went to Jim Prescott to do postdoc, and then he got the first job in Case Western, and then he moved to Vanderbilt, that everyone knows. And Chuck, I think, is unusual in that he is has a very creative mind, and he thinks broadly and generally. I mean, he always makes science look easy, but then he always addresses important problems. And every time I hear him talk, he always talk about different subjects. So, but there's a theme that's about using NMR and then membrane NMR, and then he tries to address diseases. So, has been a, you know, I think Chuck has a very successful career, and he is very good at public services also, professional services, and very good teacher. His talk is always wonderful to listen to. But I don't want to take too much of your time. It's I learned that you only have 25 minutes, so I'll just stop here.
Yeah, thank you very much for the introduction. Actually, as we have today a second introduction from Jim Prescott, so I will quickly play this. Before then, of course, so this does not go away from your 25 minutes, Chuck, so
okay.
don't worry. But,
well, it's my pleasure today to introduce to you someone I've known now for more than 30 years, Professor Charles Sanders from Vanderbilt University, or as most of us know him as Chuck. Chuck started his education at Milligan College in Tennessee. He got a BS in math and chemistry. Did his PhD at Ohio State with Mingdao Tsai. And then in 1989, moved to my lab while I was at Yale University as a postdoc, and just spent a couple of years there. But it was an exciting time, and I think that's when he became interested in membrane systems, really through working with lipid micelles, or as we now call them, bicelles. These entities that spontaneously orient in magnetic fields and allow us to measure residual dipolar coupling. And I think it was actually Chuck that actually coined the term bicep. At any rate, from there, he moved on to Case Western University in the Department of Physiology and Biophysics. Spent quite a number of years there, and then in 2002 moved to Vanderbilt, where he's been ever since. He's worked all that time really on a variety of membrane systems. Diacylglycerol kinase, PNC voltage-gated channels, amyloid precursor protein, the transmembrane parts of that. And of course, is used a good deal of NMR in those studies, but I think characteristically much more than that, including, you know, some pretty wide-ranging things. Patch clamp studies, genetics, always driven, I think, by trying to understand the functional properties of these systems. He's, of course, received several awards in the course of these studies. The Antitrace Membrane Protein Award from the Biophysics Society, the Hans Neurath Award from the Protein Society. He's done a lot of service to our community as well. Of course, many of you know him as Associate Editor for Biochemistry, which he did for more than a decade. And even now, he continues that transition. He's not just a professor at Vanderbilt, but he's also Associate Dean for research. So it really is with great pleasure that I introduce to you Chuck today to talk about some of his more recent work, actually using NMR to screen potential pharmaceutical leads that target transmembrane segments of proteins. So with that, I apologize, Chuck, that I can't be there in person today. I'm sure some members of my group here at the University of Georgia will be logging in and listening, and I can hear from them about it as soon as you're finished. So with that, I'll turn it over to Marcus, probably Marcus, and eventually to you. Thank you.
So now take it from there, Chuck, please.
So I'm actually kind of speechless and near tears. I don't know where I would be scientifically if it weren't for Ming Dao and Jim Prestigard. I just had really good fortune to get into two great labs, and I've really valued both the mentorship I received while I was in both those labs, but over the years, their support as well. And I'm really honored to be invited to speak in this series. Edition number 74. I mean, that's a remarkable track record, and I know it's a lot of work to organize something like this, so thank you very much. So I want to talk today about a project I haven't spoken on yet, and this is really the PhD work of my graduate student, Matt Castro, here, who's about to graduate in about a month from now. And you can see he's got a little model of a nanodisc here that he's made. This was early in his PhD studies. And basically what we decided to try to do was just to ask the question, if you have a sort of a simple single span membrane protein, what can you expect if you screen that protein against a library of compounds? Are you going to find anything that binds or not? And this is kind of actually an important question. This is a slide that Mac put together, and I was actually not even aware of this information. So it's great when your students dig up something that you didn't know. And this is basically just showing how important single span membrane proteins are in human life. And what you see is that, you know, in simple forms of eukaryotic life like yeast, 1% of the proteins are single span membrane proteins. But if you go to humans, it goes all the way up to almost 6%. And so there's actually over 1,200 of these proteins. And so I think that's quite impressive. And it suggests that whatever makes us human, much more complex than a yeast, is closely linked to this large family of proteins, many of which are drug targets. And so today, while I'm going to talk about the amyloid precursor protein as the model system, I'm really going to treat that as a model system representing this entire family of single span membrane proteins and not really focus so much on the Alzheimer's relatedness of this. Having said that, let me introduce the protein that we use for our studies. So of course, the amyloid precursor protein is quite a big protein, almost 700 residues. It's a single span membrane protein. And in the amyloidogenic pathway, it gets cleaved first by beta secretase just outside the membrane, and this gray domain floats away. And that leaves a 99 residue transmembrane stub that we just call C99, the 99 residue C-terminal domain of the amyloid precursor protein. So this protein itself then gets cleaved in the membrane by gamma secretase to release amyloid beta. And all of you know about amyloid beta. I'm not going to talk any more about it today. What I'm going to focus on today is C99 as a representative single span membrane protein. And what I want to know, or what we wanted to find out, was how hard would it be to screen for small molecules that can bind to such a simple single span protein. And it was not at all clear what the answer would be to this, because there's not much structure to this protein. And so we had determined the structure in detergent mice cells a number of years ago. You can see it has a sort of kink transmembrane helix. We think this is actually a flexible hinge. And there's a little amphipathic helix on the membrane surface, a disordered N-terminus, a long disordered loop on the C-terminal side, and then another So, you sort of get into questions of, just how much affinity can you get for a small molecule to just the helix, for example. And so that's sort of the question we were interested in. Now, it turns out that the cleavage, I'm going to go back one slide, the cleavage of C99 to form amyloid beta has been targeted by various drugs. Those drugs target then gamma secretase, so they inhibit gamma secretase cleavage. And I want to point out that the reason those compounds have not been successful, or one of the reasons they haven't been successful as drugs for Alzheimer's disease, is because if you inhibit cleavage of C99, you also inhibit cleavage of the Notch receptor, which is another important substrate for gamma secretase. And so one of the things that we wanted to do in our screening exercise here was see if we could find compounds that bound to C99 that don't bind to Notch, okay? And then we were gonna, if we found compounds like that, we would test them to see if those compounds inhibit cleavage of C99, but not Notch, right? And that would be, and we weren't in any way trying to develop an Alzheimer's drugs. We just wanted to see if you could do that. So that was an extra wrinkle to this project. And I should point out, Catherine Dietrich in my lab a few years ago determined the very simple structure of the transmembrane domain of Notch, as shown here on the right. So here's our basic screening funnel that we use for this project. We started with the Fezzik library. Of course, Steve Fezzik is our neighbor, and he and members of his lab both provided us with access to their small molecule library, and also gave us a lot of advice. So I want to acknowledge Steve's contribution and his help here. And so basically the initial screen was just to take these compounds and do a single point titration into to C99 in LMPG mice cells at neutral pH. And if we saw changes in the spectrum, then we would do follow-up studies on those candidate molecules. And what we did was we would then do a multi-point titration in LMPG mice cells, anionic mice cells, to look to see if we see a binding curve characteristic one-to-one binding. We would then, and if the answer was yes, we see binding, then to validate that this is a real hit, we would repeat a titration in a totally different model membrane system, DHPC, DMPC bicells, ionic bicells. And again, look to see, do we get one-to-one binding? What's the KD? And to be really thorough, we would actually then repeat again in yet another model membrane system, neutral micelles, dodecyl-molybioxide micelles, and again, look to see if we got binding. And then we would, one of the things that you really have to do anytime you do library screening is you have to confirm that the compound you think you're working with is the real compound. So you have to buy the compound from a second hardy vendor and repeat one of these experiments, make sure that it's the right compound, that it really does bind. And then we did a counter screen. We did a counter screen where we titrated the compound into notch solutions. And there the goal again is to show, in this case, that it doesn't bind to notch. We're looking for compounds that bind specifically to C99 that don't bind to another single span protein notch. Okay, so we did this with the 16,000 library, and basically we got 7 preliminary hits. And it turns out that so we ran through the validation studies, and basically that filtered out all 7 hits. So, you know, the FESIC library are molecular fragments, right? So they're not drug-sized molecules, they're like parts of drugs. And so what we learned from this is that you don't get enough affinity, there's not enough potential affinity built into those compounds to bind to a single span membrane protein that doesn't have any, you know, complicated tertiary structure where there would be a binding pocket. So that was actually interesting to learn. We also rediscovered a certain type of compound that are called PAINs. And these are, PAINs is for pan-assay interference compounds. So these are compounds that are well known to drug discoverers as compounds that tend to bind to everything and non-specifically. And so this had not been seen for membrane proteins before, but this is one of the FESIC library compounds here. And what you can see here in panel A is a titration of LMPG. So this is a TROSY spectrum, and I'm showing the superimposed spectra as we titrate from 0 to 3 millimolar compound. And it's actually remarkable how many peaks shift and how large the shifts are. So we were initially quite excited about this result. But then we crunched this through our validation system. And so one of the things that you see over here in panel C is that you don't get saturation of binding. So even at 3 millimolar, you're still seeing shifts. Okay, so this is characteristic of some sort of non-specific interaction. Very often PAINs themselves form aggregates, and it's the aggregates that are binding to your protein. We also saw that this compound also shifts many peaks in the notch spectrum, again indicating that it's not binding specifically to C99, it's just generally binding whatever protein happens to be present. So I think, you know, those of you who are thinking about doing something like this with membrane proteins, do keep in mind these PAINs compounds are sort of a well-known phenomenon in drug discovery, and they're a nuisance. It's kind of interesting to imagine whether there is some useful applications of these compounds, but I'm not aware of any so far, at least. So the FESIC library really didn't give us any positive results. So the other big thing Mac did was we reasoned, well, maybe it's just that the compounds are too small. And again, you can't get enough molecular contacts and any real, you know, interactions that really stabilize complexes. So what if we went to real drug-like molecules, which are bigger, more complicated molecules? So we next, and this is the second part of the talk really, the next thing we screened was the FDA approved drug library. So there are 1,300 compounds that here in the United States at least have been approved for use in humans. And so we did screen all 1,300 compounds, and we believe we found one bona fide hit, and it is vertiporfin. And so as you can see, it's a heme compound or a heme-like compound. And let me show you the data that we think really validates this as a true hit. So here in panel A is a titration of C99, again in LMPG micelles with vertiporfin. And we really see, under these conditions, we see two things. We see some peaks that shift. They're not very large shifts, but they are reproducible. So we've taken to do everything in triplicate. So Mac actually ran these titrations 3 times and got the same results. And so the shifts we're seeing here are real shifts. We also saw a number of peaks that broaden upon titration. They don't shift, but they broaden possibly due to motion, but also be possibly due to intermediate time scale exchange. And so in panel C here is then the actual dose response curve. And you can see these shifts fit very nicely to a one-to-one binding equation. And when you look at different peaks, they give roughly the same KD in each case. So we're seeing binding affinity of in the range of 15 micromolar for vertiporfin. To our delight, when we titrated vertiporfin into notch samples, the notch peaks don't shift. And so vertiporfin does not bind to notch. So it's binding specifically, we believe, to C99 and not to other single span membrane proteins, or certainly not to notch. We repeated these titrations in bicells. And in bicells, what you see is actually slow exchange behavior. And so if you look at this threonine 761 here, what you see is that as you do the titration, one peak disappears and another peak grows in right next to it. So slow exchange. And so you can plot the changes in peak intensity. And once again, we get a KD of around 15 micromolar, and we get that from two different peaks that we fit the data to. And so now we're seeing the same interaction in a different model membrane system, which gives us confidence that this is a real protein ligand interaction, not something that's being specifically modulated by the micelles. And once again, we saw that vertiporfin doesn't bind notch, this time in bicells. We did repeat these experiments yet again in neutral micelles, dodecylmallobiose micelles, and got the exact same results. The affinity was a little bit weaker. It was 50 micromolar, but it's not surprising to me that going from model membrane system to system, you would see modest changes in affinity, which is what we saw. So, so that really strongly suggested we had a complex. We did a couple of follow-up experiments. One is we mutated the peaks that seem to be most sensitive in terms of their resonances to vertiporfin. And so if you mutate these two residues, either of these two residues, and then repeat the titration, now vertiporfin doesn't bind with noticeable affinity. So we were able then to knock out binding using mutagenesis, which tends to confirm then that there was a direct interaction going on. We did run some NOE experiments, and the data is really ugly, but we do see NOEs between the protein and the drug. And then we did one last experiment. We were a little concerned that maybe the shifts we were seeing were not due to drug binding, but that the drug was somehow indirectly driving a change in the oligomeric state of the protein. C99 is known to form both monomers and dimers, and so it seemed feasible that this could be the case. And so we ran two different experiments to show that this was not a problem. One was we collaborated with Brandon Rutlow's lab at the University of Michigan. So he's a Carol Robinson trainee, and they do these super sophisticated, almost magical forms of mass spec. So they did native mass spec and basically showed that as you titrate vertiporfin into C99, that the protein remains mostly monomeric, and there is no, it is not driven to oligomerize. And then we also did cross-linking experiments to get the same result. Here in the left, we're titrating with the cross-linking agent. And yes, as you titrate with the cross-linking agent, you do see that you get some dimer at higher cross-linking agent concentrations. That's what you expect. But then on the right here, what we did was we kept constant cross-linking concentration and titrated in vertiporfin going across. And now you see it remains monomeric all the way through the titration. So again, we don't believe that vertiporfin is changing the oligomeric state. So based on these results, we're very confident we have a specific complex forming between vertiporfin and C99. And the affinity is not that bad, right? 15 micromolar is not a super bad affinity. It's actually okay. So we actually think that if you had a much larger library, you could find other compounds that would bind to a protein like C99. And I think that's kind of good news, actually. And so that's something we may or may not pursue in the future. Mac's getting ready to graduate, and so whether we continue this in the future or not, we'll see. And so we did do a little modeling. We took our chemical shift perturbations and used Rosetta-based modeling to come up with sort of a very crude model for the complex in my cells, and this is what we found. Now, one of the things that you'll notice is interesting about this model is that, you know, here's the drug bound to the transmembrane domain. It also has contacts with this N helix, which is in the extracellular domain. But the C terminus of the protein, which is cytosolic, because of this long floppy loop, it can actually snake around and interact with the drug as well. And so part of our affinity is coming from an interaction that can't occur in true lipid bilayers. So this is a new form of micellar artifact that we should all be aware of. And it doesn't mean that like these studies have no merit, but it does mean you have to be very careful. And we believe that, and in fact, I think Mac did this. I think he cut off that tail. We still get binding, but the binding is weaker because of the lack of this interaction. So one last experiment and then I'll be done. So we also did finally look to see, does vertiporfin specifically inhibit C99 cleavage by gamma secretase? And what does it do to Notch cleavage? And so these experiments were done by Michael Wolf, who's sort of the world's expert in gamma secretase biochemistry at the University of Kansas. And so basically, and this assay in this case is looking at the intracellular domain that's released when gamma secretase cleaves C99. And so as shown in the bottom panel here, vertiporfin does indeed cleave gamma secretase, it inhibits gamma secretase cleavage of C99. The IC50, which is not necessarily the exact same thing as the KD, is 160 micromolar, so it's a little weaker than the KD. The conditions of these assays are sort of a witch's brew. They take native membranes and add a submicellar concentration of chaps to the membranes and run the assay under those conditions. It's never been exactly clear to me what the state of the membranes are under these conditions. So I'm not surprised that IC50 doesn't match the actual KD for binding. But anyways, this we thought was a very good result. The Notch result is more complicated. And so we also look to see if Notch, what the vertiporfin does to Notch cleavage. And so at 0 Notch, you get basically 100% cleavage, the little red dot here. After you add vertiporfin, after you start titrating in vertiporfin, what you see is at low concentrations, it actually activates cleavage rather than inhibits cleavage. However, as you continue to titrate in vertiporfin, eventually you do see inhibition. So we think that this vertiporfin effect on Notch cleavage is probably not due to binding to Notch, and we know that it doesn't bind to Notch, but it probably has to do with some sort of effect on the model membranes that are being used, and or a direct effect of the compound on gamma secretase, which is sort of a notoriously sticky protease. So I'm not totally satisfied with this result, but it is the result that Mother Nature delivered to us. And so we sort of partially proved the concept that you could do this substrate specific inhibition, but only to a certain extent. And so I'll leave you with that sort of unsettling note. I just want to thank, again, my lab here on the left. And I want to thank, again, the Rutlow lab and Mike Wolf's lab. And I also can't help but plug the Protein Society Symposium in San Francisco, July 7th through 10th. Hope you can make it. And so thank you very much. Do you think I should stop?
Thank you, Jack.
Thank you. Should I stop sharing or? Okay.
Oh, it's okay. I think it's, again, very, very interesting talk from Chuck. See, I don't see questions. Could someone show me the questions? Let's see.
Please look in the Q&A.
Yeah. Q&A, yes. Okay, there's a question here. you mentioned that you saw in molecular NOEs, were these N15 labeled samples, or do you also have C13 labeled samples to see in the molecular NOEs? Do you use these for the Rosetta model? And this is from Carol Post.
Yeah, so we did like a really crude experiment. We actually just did just protonated protein and protonated drug and looked for proton-protein NOEs in a two-dimensional proton-proton nosy spectrum. The thing about this drug, the one good thing about vertiporfin is there's a number of peaks that are outside of the envelope of all the protein peaks. And so we just simply wanted to know, do you see any NOEs between the drug and the protein? And so the peaks, so we definitely saw NOEs. The peaks were not assigned, so we weren't able to use those in our modeling. Yeah.
Okay. There's a hand raised. Go ahead.
Okay.
Yeah, two more questions in the questions and answers. And
maybe I take the opportunity
let's go for
first.
those. Yeah.
Okay.
Okay. So there are Scott Prosser. Hi, Chuck. You know that what the C99 interactome looks like and the influence of this drug. APP can activate specific proteins in the brain.
Yeah.
That's a question.
Yeah, that's a good question. So let me point out that we had no intention of developing this as a drug. This is really a model system for looking at binding. So we make no pretensions in that regard. Yeah, C99 has a very complicated interactome, and its function is still not well defined. If you knock the amyloid precursor protein out of mice, the mice are a little slow and a little stupid, but other than that, they're perfectly healthy mice. And so it's really still mysterious what is the native function of the protein. And yes, it has been shown, for example, that it can activate G proteins, but it actually has a very complicated interactome, and knowing which interactions really matter is not yet clear.
Okay, thank you. There's another question from Kevin Gardner. Beautiful talk, Chuck. For your screens, at what KD do you cut off for a compound to be considered to be a hit?
Yeah, so basically we were looking for anything that would titrate with an affinity of 2 millimolar or tighter as a 1-to-1 complex. Yeah, so not a very high bar at all, right? 2 millimolar is really very weak binding, so.
Okay, so I think do we have time for another question from the panelists?
I'm sure there is time.
Go ahead, you can start the time.
So I was, thanks for the very nice presentation. I was just wondering about this Dodecil, which you used also for the, I think, uncharged or neutral mice cells. I mean, is that very common? I mean, I haven't seen that before used for NMR. So could you comment on that?
Yeah, so yeah, he's asking about dodecyl mallobioside. So actually, that's a compound that I synthesized with my own 2 hands when I was in Jim Prestigard's lab. Right. And we published an abstract on it, but we never published a paper. And so a couple of years ago, I was looking for a detergent that could be used to solubilize cholesterol. And it's very hard to do that. And I remembered this compound. And so I asked Anatrace to make it, and they now make it and market it. And it basically, it's good at solubilizing cholesterol, which is a really unique property. And it also works a little bit better than dodecyl maltoside for NMR studies of membrane proteins. So we had a biochemistry paper on it a couple of years ago. Yeah, it's an interesting compound, and you know, Jim Prestigard's idea once upon a time. So.
Thank you.
Okay, thank you. So if there's no more question, let's close this talk. Chuck, thank you again for the very wonderful speech.
My pleasure.